Good morning, everyone, and thank you for the introduction. My name is Phyllis Klarmann. I am a senior scientist and program manager for Recover at the South Florida Water Management District, working on the science of Everglades restoration. I'd like to quickly acknowledge my co-authors below, but please note that dozens of others contributed as technical experts, stakeholders, and other interested public throughout the process of updating the performance measure you will learn about during my presentation today. A quick introduction of the historic alteration of the South Florida landscape and the Comprehensive Everglades Restoration Plan, or SERP. The historical flow regime of the Everglades began north of Lake Okeechobee in the Kissimmee River and chain of lakes to where the city of Orlando is today. Water would then spill over the south rim of the lake and via sheet flow would move south into the Everglades proper all the way to the Florida Bay at the southern end of the peninsula as indicated on the left-hand side of this figure. The Central and South Florida, or CNSF project, followed major hurricanes in the late 1920s, which resulted in the deaths of thousands of people living and working south of Lake Okeechobee. The CNSF project resulted in today's expansive drainage system via a network of canals, levees, which has resulted in the loss of and compartmentalization of habitat and significantly different hydrology compared to its natural state as indicated in the center map. For the northern estuaries, these include the St. Lucie Estuary on the Atlantic coast and Caloosahatchee Estuary on the Gulf Coast of Florida. Alteration to the landscape here includes a connection to Lake Okeechobee that did not originally exist, resulting in altered volume, timing, and distribution of freshwater inflows as a mechanism for flood control, which of course have an impact on estuarine ecology. When I refer to restoration in the context of these estuaries, salinity is one primary driver of reestablishing a healthy ecosystem with a focus on reductions of deleterious freshwater inflows during the wet season and not enough base flow during the dry season. Recover, or Restoration Coordination Verification, is the multi-agency, technical, and scientific arm of the Comprehensive Everglades Restoration Plan, comprising 12 state and federal agencies, including 2 tribes. We have 3 main missions. Evaluation, in which we work with SERP project teams to evaluate and maximize the contribution made by each project to the system-wide performance of the SERP, using predictive modeling and performance measures, one of which you'll learn about today. 2, assessment, in which we measure and interpret actual responses in the natural and human systems as SERP projects are brought online, or in other words, our monitoring program. And 3, planning and integration, identifying potential improvements in the design and operation of SERP projects, consistent with plan objectives, or in other words, adaptive management. The Northern Estuaries module of Recover includes a few different ecological indicator species which are monitored as part of Recover's monitoring and assessment plan to assess SERP progress. These include the eastern oyster, as well as the submerged aquatic vegetation, or SAV community, which includes several species that run the gamut of salinity regimes, which created an interesting challenge for us in establishing freshwater inflow and salinity targets with this performance measure. Recover performance measures, as defined by the Recover Project Management Plan, are defined by indicators that are evaluated and assessed using measurable parameters with quantitative targets or assessed by trend. For the northern estuaries, because salinity is a primary driver for hydrologic restoration in SERP, we need a tool to predict how freshwater inflows to the estuaries will change, provided, for example, additional water storage. Further, we understand that our ecological indicator species require specific salinity conditions. Thus, we start by using well-established flow to salinity relationships to set environmental flow targets that will support a healthy estuarine ecosystem. Next, these environmental flow targets must then be paired with the Regional Simulation Model, or RSM. The RSM is a link node model that simulates water management operations throughout South Florida by available infrastructure, including deliveries to various users and to the environment. This model can simulate current conditions as well as projections to future conditions, which include new SERP infrastructure using a 51-year period of record of data from 1965 to 2016, which includes rainfall, reference evapotranspiration, land use land cover, public and industrial water supply, and storage volume and topography. Finally, together, these simulated model outputs from RSM for the northern estuaries include freshwater inflows in cubic feet per second, or CFS, and other measures of timing, duration, and return frequency of specific flow envelopes that result in given salinity conditions. For CERT project planning, these outputs are based on simulations of the 51 period of record, given different infrastructure and operational conditions. These outputs are compared and have ecological significance based on our salinity and flow targets, giving us a benchmark of expected project benefits. The first salinity envelope PM for the northern estuaries was developed in 2007, but flow targets for each of the two estuaries were only based on one or two indicators and only in specific locations. On the left is a map of the St. Lucie estuary, where the target salinity of 12 to 20 at the Roosevelt Bridge in the small red box on the left supports oysters and shoalgrass, more marine species, and would be met with 14-day average flows of 350 to 2,000 CFS. On the right is a map of the Caloosahatchee estuary, where salinity targets only dictated that the upper estuary, indicated by the larger red box on the right, have a salinity less than 10 to support tape grass, a freshwater or low salinity adapted SAV, and would be met with monthly average flows 450 to 2,800 CFS. Performance measures are living documents, and therefore, with newer and better ecological information or modeling tools, RPMs are meant to be updated periodically to reflect the best science available. From 2018 to 2020, we established the following goals for a salinity envelope performance measure update for the northern estuaries. This included an expanded review of the literature and our monitoring data to update salinity tolerances of our ecological indicator species. Use an estuary-wide approach, looking at salinity gradients throughout the entire estuary instead of single location targets. And finally, we wanted to identify flow envelopes that resulted in salinities that were optimum for these species. We also wanted to identify stressful and damaging conditions for each of these ecological indicator species. For the purpose of this talk, however, I will only focus on how we develop the optimal salinity envelopes and their respective flow targets. For the first step to establishing optimum salinity and flow targets, we did an updated literature review, which included synthesis of field observational and experimental studies, lab experimental studies, monitoring data, and regional subject expert opinion. Each of the ecological indicators were given an optimum salinity range included in the red box in the table below. Optimum is defined as the conditions yielding the highest or best response to monitoring variables such as growth, density, photosynthetic capacity, among others. Step 2 included employing a hydrodynamic salinity model to update the established flow salinity relationships I discussed earlier. The curvilinear hydrodynamic three-dimensional model, or CH3D, is based on more than 10 years of measured inflow and observed salinities from multiple water quality stations in both estuaries. The correlation between observed and modeled salinity, which includes boundary conditions of tide, gauged inflow at the structures, modeled tidal basin runoff, and observed wind at the water surface, has an R-squared of greater than 0.9 for both estuaries. A given flow range or envelope was simulated using CH3D, for example, 450 to 2,800 CFS for the Caloosahatchee depicted right, the outputs of which were used to calculate average potential habitat area. Potential habitat area is defined as the average area where salinity is within the optimum range for a specific ecological indicator for a 14-day period within each model grid. One product includes the map on the right with modeled average salinity throughout the entire Caloosahatchee estuary from which average potential habitat area is calculated. Step 3 included performing a sensitivity analysis of the calculated potential habitat area of different flow envelopes for each ecological indicator species, and we plotted them as line graphs relative to the 2007 targets for comparison. Here is an example for shoalgrass, a marine SAV in the Caloosahatchee estuary. The lines represent the low flow bound of a given flow envelope from 450 to 750 CFS. The x-axis includes the high flow boundary of a given flow envelope from 1,800 to 3,000 CFS. So for example, the red dot centered in the graph represents 450 CFS at 2,800 CFS on the x-axis, which was our 2007 target for the Caloosahatchee, 450 to 2,800 CFS. At this location on the y-axis, we have one with the other flow envelopes measured relative to that performance. The y-axis above 1 is in 5% increments better than the 2007 target, while those below 1 are in 5% increments worse than the 2007 target in total area, where the optimum salinity is 15 to 45 for shoal grass for a 14-day period. We then compared the Caloosahatchee graphs for each of the indicator species and used best professional judgment to select a few flow envelopes that both perform better than the 2007 target, which are above the red dash line or above 1 on this graph, and maximizes the benefits across all species. Again, a challenge given they each have different salinity optima. The last step included the generation of salinity maps by plugging in the smaller selection of proposed flow envelopes to compare to the 2007 targets. These maps, for example, include average salinity here on the left in the yellows, greens, and blues, and percentage of time within the optimum salinity envelope on the right in the yellows, oranges, and reds. These two maps are the average salinity and percentage of time within the optimum envelope for tape grass, which requires salinity less than 10, based on, for example, depicted here is simulated flow 750 to 2100 CFS. Once again, we compared all salinity maps for each indicator and each estuary to determine which flow envelopes performed best for all indicator species. Here are the final optimum flows for the St. Lucie and Caloosahatchee estuaries compared to their 2007 targets in the red box in the table above. For both estuaries, the flow envelope narrowed considerably. For the St. Lucie, the low flow and high flow boundary decreased in order to produce optimum salinities, namely for oysters and the submerged aquatic vegetation community, and expected to minimize the deleterious high inflows that often result in depressed salinities, mass oyster die-offs, and typically minimal SAV present in the St. Lucie. For the Caloosahatchee, the low flow bound increased from 450 to 750 CFS to improve salinity on the lower end for tape grass in the upper estuary, and the high flow bound decreased from 2800 to 2100. And this was meant to prevent stressful and damaging low salinity downstream that could impact oysters and marine SAV. Since the update of this performance measure, we have been able to test it out for the evaluation of the U.S. Army Corps of Engineers Lake Okeechobee System Operating Manual, or LOSOM. While the Recover PM is meant to be a restoration-based evaluation tool, it has been incredibly useful to compare regulatory releases from the lake which impact the estuaries. These alternative operations are modeled using the RSM, just as it is in CERT planning, and results in model outputs like those you see below, comparing optimum stressful and damaging flow events to the northern estuaries based on some existing condition base reflective of today, a future date in which no operational changes are made, and several operational alternatives. While LOSOM is currently entering its third iteration, where a selected operational alternative has been selected and will further be optimized, a significant takeaway from using our Recover performance measure and a number of other performance metrics for LOSOM is evident. Water management operations are limited by the infrastructure available in which to send water, and thus, additional storage capacity and other features of CERT projects is key to system-wide restoration of the Everglades. Finally, this performance measure will also be used in future CERT planning studies. With that, I'm happy to take any questions. If there's time, you can also download the Recover Salinity Envelope PM at the bit.ly link below. And I encourage you all to attend NCER Session 29 on Tuesday, August 5th, to learn more about Recover Science.